So get started answering a question that was in the Slack that I missed at the very beginning, which is from Taylor, who asked if there's some benefits specifically for viruses that interact with the renal angiotensin system. And so that's the system that ACE2 is part of. And I think it's a really great question that we still don't have an answer for. There's been a lot of provocative papers that have suggested that this could be important. So for those of you that are unfamiliar with the renal angiotensin system, it's quite complex and regulates a lot of fundamental physiology. It's what's targeted by, for instance, a lot of your blood pressure medications, but it also feeds back on the immune system as well. And in particular, ACE2 is a receptor for compounds that regulate inflammatory responses. And so the virus, by binding ACE2, likely causes a reduction in its expression level, because when the virus binds, it causes the receptor to be taken inside the cell. And so it shifts the way that system is working systemically. And so again, there's been a lot of speculation that this might have something to do with a lot of the strange symptoms that people with SARS-CoV-2 have reported that are separate from the respiratory symptoms. But nothing really definitively has been shown about that. And probably one of the major things that's limiting our understanding of that is that the mouse models for SARS-CoV-2 are still really, really poor. And part of this is because the ACE2 in the mouse did not natively bind the spike protein from the original virus strains. Interestingly, as the virus has evolved, and so the most recent variants of the virus actually intrinsically infect mice now that didn't before, and it's because they bind the mouse ACE2. One of the things that's evolved in the variants is a much tighter binding to ACE2 than in the original strains. So it's suggested that at least the strength of binding to ACE2 is something that's being selected for, and whether or not that's actually related at all to the ability of the virus to regulate ACE2 expression levels is, I think, a really interesting question.
That's really interesting. Thank you.
Are there other questions, or I can go through the variant slides. If there are questions, you could just unmute yourself and ask them.
For the spike protein on SARS-CoV, has anybody looked at breaking down the peptide chains and looking at responses for T cells in like shortish chains, like different peptide pools?
Yeah, sure. Give me 1 second here, and I will show you data on what that looks like. That is a great question. So yes, that has been done, and it's been done for spike more than for any other protein. And there's good reason for that. So one reason for that is that spike is the only protein that's in the vaccine. So if you're just trying to study vaccine responses, then that's what you're going to study. And then people have been interested in it because spike is mutating the fastest in terms of the variant response as well. And so that's another reason why it's been looked at. But if you just restrict yourself to the spike, you're missing, 90% of the virus plus. And so, so that's been an issue is there's this trade-off of, do you look really intensely and well at one protein, or do you try to look at all the proteins at once? And that's kind of the trade-off I was talking about earlier, where I think we're not necessarily estimating the correct amount of CD8 T cell responses. And so give me 1 more second. I'm going to pull up, I don't have these as slides, but I'll put some references for you in the chat afterwards that show this. Let's see. It's a good one here. So there's a group out of La Jolla that's been doing a lot of the, here's a good one, okay. Has been doing a lot of the work on the peptide pools. Just for practical purposes, for those of you that don't work directly in bench science, another limitation of this peptide pool approach is that peptides are actually kind of expensive. So like one good peptide made at a high quality from a standard vendor is like $200 or $300. To make the entire proteome of SARS-CoV-2 in the way that it's used to study responses in people was cost about $120,000. And that's a lot of money for one lab to take on. And so the labs that have done it have usually had some special deal, or they're labs that actually make their own peptides in some way. So let me show you. I'm going to just share my screen here. Share screen 2. Are you guys seeing like a graph and my mouse moving around? Yeah, okay. So this is this paper here. It's on the bioRxiv, and it's now published, I think, in Cell Reports Medicine. And so what they were looking at was, if we take the spike protein and take people that have been vaccinated and look at the mutations in the spike protein that correspond to the variants, do we see changes in the immune response with the variants? And their conclusion was, no, we don't, sort of on a population level. But this is sort of the way they measured it. Is it's not really an ELISpot, but it's a functional assay like the ELISpot, where they take CD4 T cells here or CD8 T cells from the blood of a person, and they just hit them with the various peptide pools of spike across these different variants. And for a few people, they did the mutations that were not in the spike. And what you'll see is, I think you can kind of get a sense of it from, if you look down here, particularly where they're looking across the proteome. This is the CD4 T cell responses, and they're using a flow cytometry-based assay to look for cellular activation. And so they look for the upregulation of activation markers on cells in response to being stimulated with those peptides. And if it goes up by a certain amount, they say the cell that upregulated that is specific for one of those peptides. So one of those small pieces of spike or these other genes. And if you look overall, there's higher levels, if you kind of squint and look back and forth really fast, of CD4 responses to CD8 responses. And that's been a consistent finding in this assay. And it gets back to the point about somebody asking is about the CD8 response being weak. This is the data that's really driven that idea that the CD8 response is weak, because it's smaller than the CD4 response. And that's the opposite of almost every other virus that you look at. Usually the CD8 response is much bigger than the CD4 response. And you can think of that, it makes sense. How do you need one quarterback, you need a whole bunch of other players. And the CD8 cells are really your other players. And so one interpretation is that this is just how it is. It's something weird about SARS-CoV-2. There's biology here we don't understand. Another interpretation is that maybe this assay is not very good. And it's less sensitive for CD8 T cell responses. And I think there's good hints of this in the literature, particularly in the literature that uses this assay. You can kind of see it here. It's a small example, but there's maybe only 10, maybe even less than 10 individuals on this graph here. And one of them had literally no CD8 T cell response that was detectable. And this is a routine finding from this group and other groups, that actually up to 50% of the people that they assay with this assay have no detectable CD8 T cell response. And you can either interpret that as people aren't making a CD8 T cell response and it's really weak, or your assay is really not sensitive. And we can kind of get at that a little bit by using another method to look at CD8 T cell responses. And so I'm going to shamelessly show you some data from my own paper. Give me 1 second. I have to switch what I'm sharing. This is also on the bioRxiv, or will be in a day or two. But it's another method for looking at T cell responses. And I'll reshare my screen here. And it's a way of looking at T cell responses that doesn't require the cell to do anything. And so ELISpots. There's an assay called intracellular cytokine staining. The assay we were just looking at is sometimes called the AIM assay. These are all functional assays where you hit the cell with the antigen and you ask it to do something to reveal that it is actually a specific cell. There's another way of enumerating T cells, which is what you want to do when you're modeling. And that's actually using a reagent that's variously called a tetramer or a multimer or a dextramer. You'll hear it called various ways, but all it is, as you can see here, you guys can see my screen now and my mouse moving around. Yeah, okay. Is the MHC molecule with the peptide of interest bound into it. And then you multimerize that in some way. And here we've stitched it to a dextran molecule, and that's why this is called a dextramer. And then you put some fluorescent probes on there so that you can use various methods to pull those cells out and count them. And so what we're looking at here is we're looking for cells with T cell receptors that will bind specifically to this peptide MHC combination, which is the target of a T cell receptor. And so this is a really nice and exquisitely sensitive way. You can go down to one cell with this approach and find T cells that are responsive to these tetramers and dextramers. And so you might say, well, why doesn't everybody do this? This is a great way of doing it. And the problem with this is that you have to know exactly what the target is. So like you have to know the peptide you're talking about. And you can't be off by even a single amino acid. You have to say that this is that peptide bound to a specific MHC molecule. And you have to know the MHC molecule. And the trouble with knowing the MHC molecule that you're going to look at is that the MHC is the most diverse locus in the entire human genome. So we all have different MHC molecules. Some of us might have some molecules that are shared, but probably other than your twin sister or brother or a statistically improbable sibling, you're not matching with another person perfectly, except very, very, very rarely. In one of the introductions, someone mentioned being a bone marrow match. The reason there's this bone marrow registry is you're trying to find somebody that you match with perfectly, or at least semi-perfectly on all your MHC molecules. So this was the list of MHC molecules. This is how we name MHC molecules in the human. Here are these letter number combinations. And then the peptides that were associated with those MHC molecules that we looked at in this study. So it was about 18, looking at various antigens. This is just scratching the surface of the potential immune response. On the flip side, we are not asking the cells to do anything. And so we're probably detecting the cells at a much higher rate than you would from a functional assay where the cell has to do something in a dish after it's been violently ripped out of a person's bloodstream. And so you gain in sensitivity in one way, but you lose in sensitivity in another way. But I'll show you. This is sort of summary data of all of these different tetramers across the individuals where they had the matching MHC molecules that would allow us to look for these immune responses in them. So these were all either infected or vaccinated individuals, and every single one of them had a really robust CD8 T cell response. Even just looking at relatively small numbers of potential antigens, we could always find CD8 T cells for virtually all of the antigens in all of the people where such a response could have been mounted because they had the right MHC response. This is just sort of looking at people after infection, after vaccination and infection, or just after vaccination. And basically everybody made the same amount of CD8 T cells if they'd been exposed to spike antigen, regardless of how they were exposed to spike antigen. That also tells you how good these vaccines are, because it's very unusual that a vaccine is as good as an infection. These were all RNA vaccines. So Macaulay, your question was, do you ever just look at the peptides? This is how you look at the peptides, but you have to go through this process of knowing exactly what peptide binds to which HLA molecule. And it's not trivial, and it takes a lot of time. And so that's why this paper that we just wrote and submitted, because it took us this long to kind of get this many well-mapped epitopes to really to look at in response. Are there other questions? If you do have a question, feel free to unmute yourself.
Yes, I had a question regarding to the immunity, long-lasting immunity between natural infection and vaccine-induced. Are there any differences?
It's a really great question. The data are starting to come out. So if you think about what data we have people that were infected for, I assume you're asking specifically about SARS-CoV-2. We have people that were infected in January, February last year that now are 15, 16 months out. And then for vaccination, we have people, for the RNA vaccines starting around December, January, so they're only 6 or 7 months out. So this is really the critical period where we're going to know what that longevity looks like. So far, it looks pretty promising. So one of the ways that you can get a sense of how promising it looks is there are data now from that same group that I showed you the bone marrow data from. They published a paper looking actually at human lymph nodes, where they did a biopsy of human lymph nodes of people that got the vaccine at various time points after getting the vaccine. And they looked at the B cell responses in those lymph nodes, and they were really big. And the bigger the response, usually the longer it lasts, because you kind of go up to a high level and then you start declining at a slow, a slow kind of constant slope. And so if you start up higher, you have a lot longer on that slope. And the cells looked by all of our phenotypic and activation markers like cells you would see in an infection. So the idea is that they might be well matched to an infection. And the paper I was just showing you, when we looked phenotypically at the CD8 T cells, again, we actually couldn't really distinguish really robustly cells that were elicited by a vaccine and from an infection. And again, that's very unusual. This is specifically for RNA vaccines. In contrast, the flu vaccine is known to not be to not induce long responses. So there's great data on this that basically within a year, you've lost the boost you get from the flu vaccine, which is another reason why you get it every year. It's probably not totally lost. It's probably just dropping back down to this sort of set point baseline level. But it's so far, the SARS-CoV-2 data is looking much better than that. There's a lot of pressure right now on Pfizer and Moderna to start releasing some of their data, because they actually have older data than anybody, because they have the people that were in their phase 3 trial. And so they probably know more than the rest of us know, because we only have access to people that were vaccinated after the trial. But it'll be really interesting to see if they come out with anything soon.
Thank you.
Any other questions?
Regarding to the last question, how long do you think that the immunity will last? Because in the plot, I've seen that the plot was until 120 days. But normally, do they predict that every year we will have to get a booster dose, or?
So this is going to be, a big topic of discussion, and it's a bit unfortunate that I think this area has become so political, because there's probably not going to be a very clear answer for a while, and that's going to lead people to making all sorts of claims probably about why there's not a clear answer. To give you the very best vaccine that we have for anything is probably the yellow fever vaccine, which was invented many, many, many years ago. And the yellow fever vaccine is actually an attenuated form of yellow fever virus. So you actually, when you get the yellow fever vaccine, you're actually getting infected with yellow fever virus. And so if we think about the principles that we've been talking about the last 2 days, which is with a vaccine, you're trying to trick the immune system into thinking it's having a normal immune response. The best way to do that is actually just give yourself the infection in some form, because you get all of the little pieces, right? You get the innate response, you get the unfolding of the adaptive immune response on the right time scale. That really only works though if you can give the infection safely. And so we haven't developed an attenuated vaccine response for decades, because it's very hard to do that in a safe manner and to get the testing done in a way that you wouldn't cause probably some adverse events. So all that said, we're probably never going to have another, I would guess, attenuated vaccine anytime soon. But we do have this one that, because of historical norms being very different, was tested and was determined to be safe. That vaccine gives you lifelong immunity. And so there are really cool studies of people that either got yellow fever or got yellow fever vaccine in like the 1920s that were still alive in the early 2000s. And they had their bone marrow looked at, and you could find long-lived plasma cells still making antibodies to yellow fever, and they had not been exposed for the last 80 years. And so that was just one exposure led them to have this lifetime of immunity. Same thing with their CD8 T cells, long exposure leads to this lifetime of immunity. And so I suspect that there's some people that have been vaccinated and or infected that will have some form of immunity for their whole life. And so the questions that then remain are, is it enough? Like, do you need a booster just to kind of raise that set point for a period of time? Does the vaccine mutate, or sorry, does the virus mutate to a point that the vaccine's no longer useful? And then who are the people that that's happened to that are making the great response, and are there people that are making a not great response? If you look at, I'm going to share my screen real briefly. If you look at the, I know I'm not in presenter mode, but it'll be a bit easier to look at this. So this is data from vaccinees looking at just antibodies. And you look at the spread of responses here. This is a little bit of a complicated study because they were looking in people that had either been infected or not been infected. But the main point I want to make is that you get this multi-log variation in individuals in terms of their response. And so from a modeling perspective, from an epidemiological perspective, from a public health perspective, do you come up with a system where you try to figure out who these people here are down here that aren't making very good responses and they need the booster? That's tough to do because you've got to get people to go in for a test. You've got to validate that test. What's a protective level? We're still arguing about that as a field. Or do you just make a recommendation for everybody, which is kind of what we do for flu, and just say, we're just gonna have a booster every year or every 2 years or something like that, and try to move, a certain target threshold of people, 70% of people, 80% of people, to where they have some target level that you think is probably protective. And I think these are going to be raging debates. I think, the recent report that Pfizer was applying for FDA approval for a booster shot shows some of the difficulty with this, because Pfizer is not an unmotivated party here. They would love for people to have to pay for a booster shot every year, or the government to pay for a booster shot every year. And so, everybody's going to come into this with some sort of bias. And right now, I don't think we have the data to say what the right answer is. The data that we need, that might take a little while to get, is what's actually a true correlative protection. So we have correlates of protection in flu. And by that, I mean, we have a number that we can measure in the blood of antibodies, and we know how many people are protected from flu if they have that number of antibodies. We don't actually really have that for SARS-CoV-2. We have some correlations that have look like they hold up pretty well, where we say, if most people who were vaccinated had this level of antibody, most people who were vaccinated are not getting infected. If you have that level of antibody, you're probably not going to get infected. But we don't really have a true correlate of protection where we know in a sort of prospective way that people above this line had this risk and people below this line had a different risk. That was a long-winded answer, touching on various things, but the answer is it could probably last anywhere from a few months to a lifetime, depending on who you are and depending on what the virus does. And it's going to be a question of where's the sort of mode of the population and what we need to do in terms of a booster in that regard.
Thank you.
I have a question sort of following up on that. What can we do as far as like modeling potential, what the correlative protection could be with the existing data? Obviously, it might take more time to have all of the definitive data on like what is an established correlative protection, but could you speak on like sort of how we can leverage the data that we do have to try to start to establish?
Yeah, it's a really great question. Unfortunately, the data we have are the data that are easiest to produce, which are these types of antibody data. And so this is easy to produce because you can get it out of serum, which is a very easy sample to take. And the assay itself is pretty easy, and it can be done high throughput. And so just like for flu, actually, our correlates of protection are generally associated with things that we can measure, but they're not necessarily mechanisms of protection. They're just good correlates of protection. But in terms of finding something like that, that's just a true correlate of protection, looking at good epidemiological studies of people that are being longitudinally followed and then get infected, but have been immune profiled prior to that infection is really the holy grail of what's going to give you a good correlate of protection, or inversely a correlate of risk. It's kind of, in a way, much easier to find a correlate of risk because you know that that person was infected. And so you know that whatever immune profile they had failed. And so if you can start finding correlates of all the people that fail, that's going to be really useful. Those studies are starting to come out because those cohorts exist now. People have been vaccinated and are being followed for a while. We're starting to see these breakthrough infections. What you're going to want to correct for with that is, what variants are around. This is also something when you're looking at the relative efficacies of the different vaccine platforms, you have to look at where they were tested and when they were tested. So Pfizer and Moderna got tested before many of the variants were really widespread. Novavax, AstraZeneca to some extent, J&J were in the field when the variants were coming up that had actually started to escape them. So this doesn't include the most recent variants, Delta and Lambda. The nomenclature for SARS-CoV-2 keeps changing in terms of what we consider a good nomenclature, but this is the Alpha variant. This is the, I actually get these two reversed, Beta and Gamma. I can't remember. This used to be the UK variant. This was the South Africa variant. This was the Brazil variant. And if you look at the efficacy of this is the Oxford vaccine, AstraZeneca, and against B1351, which is the South Africa variant, it was very, very poor. So you think about the lines that I showed you up here and how beautiful that was and how exciting. The lines are superimposable. And so that's something I think in the modeling that actually modeling is going to be really critical for is adding into a model of protection antigenic distance.
I have a question.
Sure.
Okay, my question is, does developing a vaccine that contains a spike gene in addition to another structure gene gonna enhance the efficiency of the vaccine, or there will be no impact at all?
I think this is a fantastic question, and I wish we had all of these answers already. Let me stop sharing. There is a very interesting paper on the bioRxiv. Let me see if I can find it really quickly. Did I find it? That actually is a nice paper, but it's not the one I was looking for. I will find this before our next session and post it, but I'll just tell you what the data were. So this was a trial of healthcare workers where they were monitoring their immune responses very closely and whether or not they got SARS-CoV-2. And what they found was that, this was very early on in the pandemic, and a bunch of people got SARS-CoV-2, but a bunch of people in their cohort never got SARS-CoV-2, and they were monitoring them weekly, regardless of symptoms, with a nose swab to look for the virus. And then they also were monitoring them with antibody tests. And there's this group of people that never developed antibody, and they never developed a nose swab positive test, but they had this big boost in their CD8 T cell responses and CD4 T cell responses to some of the non-structural proteins, and particularly the non-structural protein NSP12. And there's this one paper, but there's probably a half a dozen other papers that have started making this claim that there are people that have been exposed to the virus and basically fought it off without the virus ever producing enough antigen to actually mount a new B cell response. And so they were purely able to control the virus with a T cell response. And so you might say, well, why do they have enough antigen to mount a T cell response, but not a B cell response? So the argument is that these T cells that they were using to clear the virus were T cells from other coronavirus infections. So the internal genes of a lot of coronaviruses are a lot more similar to each other than the external genes. True in a lot of virus families. It's definitely true for flu. So the internal genes kind of don't have a lot of wiggle room because they have to do a lot of functional work. And so they can't mutate as nearly as much as the surface proteins mutate. And so we all have had other coronaviruses, they're common cold viruses. And so we all have T cells to these other common cold coronaviruses. And the argument of a series of these papers is that if you have enough of those T cells, that's actually what's holding you back from getting really sick. That's why in many, many people, it's a very mild infection, even though in principle, we haven't seen it before. And it's actually one of the arguments for why in kids in particular, it's very mild, because kids get common cold coronaviruses all the time. And so they maybe have boosted up these T cell levels. This hasn't been, there's correlations that would support this, but it definitely hasn't been proven statistically. But the argument from those papers is we should start including internal genes in the vaccines because we'll make really good T cell responses to these. And those T cell responses will help boost and control the infection. Even if say the spike protein drifts really far away, we'll have this other protection coming from these internal protein T cell responses that are much, much less likely to drift.